Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1995 witnessed a singular achievement in the burgeoning discipline of genomics, the complete sequencing of the bacterium Haemophilus influenzae RD, accomplished through a revolutionary approach pioneered by Craig Venter, Hamilton Smith, and Claire Fraser at the Institute for Genomic Research, later known as TIGER, is undertaking, detailed in their seminal publication Random Sequencing and Assembly of Haemophilus influenzae RD, transcended a mere cataloguing of genetic information. It represented a paradigm shift in biological investigation. Its impact resonated far beyond the walls of TIGER, influencing the trajectory of genome research for decades to come. To fully appreciate the magnitude of their accomplishment, one must consider the technological and conceptual constraints that defined the field at that juncture. Traditional sequencing methodologies, reliant on hierarchical, clone-based approaches, were painstakingly slow and laborious. The process demanded isolating large DNA fragments, meticulously constructing overlapping clones within vectors such as plasmids or bacterial artificial chromosomes, and then sequencing these defined fragments individually. This process, while accurate, proved exceedingly time-consuming and expensive, effectively restricting whole genome sequencing to the simplest of organisms. Venter, Smith, and Fraser, however, adopted a radically different strategy, now commonly known as whole genome shotgun sequencing. Diverging from the prevailing wisdom, they eschewed the traditional reliance on physical maps and clone-based approaches. Instead, they shared the entire Haemophilus influenzae RD genome into millions of small, randomly sized fragments, typically between 1,600 and 2,000 base pairs. These fragments were then cloned into plasmids, and a relatively small amount of sequence data, on average 500 base pairs, was generated from each end of each plasmid clone. The subsequent computational challenge involved assembling these vast quantities of short, unordered sequence reads into a contiguous representation of the entire genome. The critical innovation lay in the development and application of sophisticated algorithms and computational infrastructure capable of managing and processing the immense volume of data. The TIGER Assembler, a software package written by Granger Sutton, Stephen White, and others at TIGER, served as the linchpin of this process. This program employed algorithms rooted in graph theory, specifically overlap-layered consensus methods, to identify and merge overlapping sequence reads, ultimately constructing longer contiguous sequences, known as contigs. The initial assembly yielded 140 contigs, which then required further analysis and manual curation to resolve ambiguities and close gaps. The Haemophilus influenzae RD genome, as revealed by this effort, comprised 1,830,137 base pairs, encoding 1,740 functional genes. The analysis unveiled a wealth of previously unknown information regarding the bacterium's metabolic pathways, virulence factors, and evolutionary history. Of particular note was the identification of numerous genes involved in DNA repair, recombination, and transposition, providing insights into the genomic plasticity of this organism. Furthermore, the annotation of the genome revealed a substantial number of genes with no discernible homology to known proteins, highlighting the vastness of unexplored biological territory. The direct applications of the Haemophilus influenzae RD genome sequence were immediate and profound. Prior to this achievement, researchers studying this bacterium were limited to investigating individual genes or proteins in isolation. The availability of the complete genome sequence provided a holistic view of the organism's genetic makeup, enabling systems-level approaches to understanding its biology. For example, scientists could now study gene expression patterns across the entire genome in response to different environmental stimuli, providing insights into the bacterium's adaptive mechanisms. The complete genome sequence also facilitated the development of novel diagnostic tools and therapeutic strategies. Knowledge of the bacterium's unique genetic signatures allowed for the design of highly specific polymerase chain reaction, or PCR, assays for rapid and accurate detection. Moreover, the identification of essential genes provided potential targets for the development of new antibiotics. By targeting these genes with specifically designed drugs, it might be possible to disrupt critical cellular processes and effectively combat Haemophilus influenzae infections. Beyond its immediate applications, the Haemophilus influenzae RD sequencing project served as a proof of concept for the whole genome shotgun approach, thereby paving the way for the sequencing of more complex genomes. Notably, it directly spurred the subsequent sequencing of Mycoplasma genitalium in 1995, also led by Venter and Fraser, and then, more significantly, the human genome. The success of these projects demonstrated the scalability and efficiency of the whole genome shotgun method, solidifying its position as a dominant strategy in genomic research. The influence of the Haemophilus influenzae RD project extended beyond its methodological contributions. It also served as a catalyst for the development of new technologies and computational tools. The sheer volume of data generated by the project demanded significant advances in DNA sequencing technology, bioinformatics algorithms, and database management systems. These advances, in turn, fueled the rapid growth of the genomics industry, leading to the development of faster, cheaper, and more accurate sequencing platforms. It is worth acknowledging the initial skepticism and debate that surrounded the whole genome shotgun approach. Many researchers, accustomed to the rigor and control afforded by traditional clone-based methods, voiced concerns about the accuracy and reliability of the new approach. They questioned whether it would be possible to accurately assemble a complete genome from millions of short, unordered sequence reads, particularly in the presence of repetitive DNA sequences. These criticisms, while valid, ultimately proved to be unfounded. Subsequent validation studies, including comparisons with traditional mapping data, demonstrated the high accuracy of the Haemophilus influenzae RD genome sequence. Furthermore, the development of improved assembly algorithms and computational tools addressed many of the initial concerns regarding the management and analysis of larger-scale sequence data. The reception of the Haemophilus influenzae RD sequencing project within the scientific community was overwhelmingly positive, solidifying the careers, reputations, and legacies of its principal investigators. The accomplishment was hailed as a major breakthrough in genomics, representing a significant step forward in our understanding of life at the molecular level. It ushered in a new era of genome-enabled biology, transforming the way researchers studied organisms, diseases, and evolutionary processes. The sheer audacity of the approach, coupled with its demonstrable success, garnered considerable attention, even from the usually reticent scientific journals. The influence of the Haemophilus influenzae RD project on subsequent research is difficult to overstate. It directly influenced the design and execution of numerous genome sequencing projects, including the sequencing of other bacterial genomes, as well as the genomes of plants, animals, and humans. The whole genome shotgun approach became the standard method for sequencing complex genomes, driving down the cost of sequencing and accelerating the pace of discovery. For example, the sequencing of the fruit fly Drosophila melanogaster, published in 2000, relied heavily on the whole genome shotgun approach, demonstrating its applicability to larger and more complex genomes. Similarly, the Human Genome Project, a landmark achievement in scientific history, utilized a combination of whole genome shotgun sequencing and clone-based sequencing to produce a complete map of the human genome. Furthermore, the Haemophilus influenzae RD project paved the way for the development of personalized medicine. The ability to rapidly and affordably sequence individual genomes has opened up new possibilities for tailoring medical treatments to the specific genetic makeup of each patient. This approach, known as pharmacogenomics, holds the promise of improving the efficacy of drugs and minimizing adverse side effects. The work of Venter, Smith, and Fraser also had a profound impact on the field of synthetic biology. By demonstrating the feasibility of assembling a complete genome from scratch, they laid the groundwork for the creation of artificial organisms with novel functions. This field, still in its early stages, has the potential to revolutionize medicine, agriculture, and industry, enabling the design and construction of organisms with tailored It is interesting to note that the pursuit of whole genome sequencing, despite the initial skepticism, ultimately proved to be a more cost-effective and efficient approach than the painstakingly slow methods that dominated the field previously. This highlights the importance of challenging conventional wisdom and embracing innovative technologies in scientific research. The legacy of the Haemophilus influenzae RD sequencing project continues to shape the field of genomics today. The methods, technologies, and computational tools developed during this project have become integral to modern biological research. The insights gained from the project have advanced our understanding of bacterial evolution, pathogenesis, and antibiotic resistance. The availability of complete genome sequences has transformed numerous fields of biology, from molecular biology and genetics to evolutionary biology and ecology. Researchers can now study the genetic basis of complex traits, trace the evolutionary history of organisms, and explore the interactions between organisms and their environment. In conclusion, the 1995 publication by Craig Venter, Hamilton Smith, and Claire Fraser, detailing the random sequencing and assembly of Haemophilus influenzae RD, stands as a pivotal moment in the history of genomics. It marked the triumph of a bold and innovative approach over traditional methodologies, ushering in a new era of genome-enabled biology. The project not only provided valuable insights into the biology of Haemophilus influenzae, but also paved the way for the sequencing of more complex genomes and the development of new technologies and applications. Its influence continues to resonate throughout the scientific community, shaping the direction of biological research for decades to come. Its contribution remains a testament to the power of innovation and the transformative potential of genomics. The ripple effects of this single paper continue to expand, influencing fields from personalized medicine to synthetic biology, solidifying its place as a cornerstone of modern biological research and a defining moment in the history of science. The meticulous work they did, even when faced with doubt, has become the bedrock for much modern genomics research. The team of Venter, Smith, and Fraser demonstrated that bold, innovative approaches, even when met with skepticism, can revolutionize scientific fields. Their work continues to inspire researchers to push the boundaries of knowledge and explore new frontiers in biology. The complete sequencing of Haemophilus influenzae (RD) serves not only as a testament to their scientific ingenuity, but also as a reminder of the profound impact that a single, well-executed project can have on the course of scientific history. The implications of this single research effort continue to unfold, cementing its place as a foundational work in the ever-evolving landscape of genomics and beyond. It truly stands as a landmark achievement. Finally, the success of this project underscored the critical role of interdisciplinary collaboration in scientific discovery. The project brought together experts in molecular biology, genetics, computer science, and engineering, demonstrating that complex scientific challenges often require the combined expertise of multiple disciplines. This collaborative spirit continues to drive innovation in the field of genomics, fostering the development of new technologies and approaches that push the boundaries of our understanding of life at the molecular level. It is a lesson that continues to resonate within the scientific community.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.